Okay, we can get started. Hello, I'm Dr. Sang Park, the senior scientist with the Institute of Cannabis Research, hosted at Colorado State University, Pueblo. With the emerging request for information on the advances being made in cannabis cultivation and risk management, the ICR is launching a monthly cannabis cultivation webinar series. The webinar series aims to provide recent updates on cannabis farming technology, cultivation challenges, and entrepreneurship, as well as a networking opportunity. We are pleased to have the Volcani Center, the agricultural research organization in Israel, as our co-host for this webinar series and co-chair for the ICR Hemp Farmers Association, Dr. Nirit Bernstein, as our presenter for our inaugural webinar in this series. Before we get started with the presentation, I would like to discuss some information about the logistics of the webinar. Today's presentation will be followed by a short question and answer period. To ask questions, please do so using the Q&A feature in Zoom toolbar. You can enter questions at any time during the presentations. We will address as many questions as time allows until the end of the hour. Please do not use the chat function to post questions for the Q&A period. Please reserve use of the chat function for technical issues and other questions not related to the presentation. Both the Q&A and chat functions can be found in the toolbar on your Zoom screen. It is now my pleasure to introduce Dr. Chad Kinney, the director of the ICR, who will introduce today's presenter and as well as moderating this webinar. Chad.
Thank you, Dr. Park, and thank you to everybody for joining the ICR and the Volcani Center for this inaugural edition of this new webinar series. It's my pleasure to introduce Dr. Nirit Bernstein as the presenter for today's webinar. Dr. Bernstein is a senior researcher and head of the Medical Cannabis Research Lab at the Volcani Center for Agricultural Research in Israel. Dr. Bernstein is a leader in medical cannabis research and irrigation with recycled water in Israel. She earned a Bachelor's of Science in Agricultural Sciences from the Hebrew University in Jerusalem and earned a Ph.D. in Plant Physiology from the University of California, Davis. Her research focuses on the production of physiology of medicinal cannabis by manipulation of environmental treatments, optimization of agrotechniques for environmentally stressed plants, mineral nutrition of plants, and plant stress physiology. Dr. Bernstein serves as a member of the editorial board of 5 international scientific journals, including serving as an associate editor for the Journal of Cannabis Research, which happens to be the official journal of the ICR. She also serves as the chief editor of scientific manuscripts in Hebrew for the Ministry of Agriculture in Israel, and has been a project leader of 60 national and international research projects. Dr. Bernstein was the first agricultural researcher to be licensed to study medical cannabis in Israel. And finally, as Dr. Park mentioned, Dr. Bernstein will be collaborating with Dr. Park as the organizing chairs of this webinar series. Dr. Bernstein, we are very pleased to have you with us today, and I will turn things over to you for your presentation. Thank you.
Thank you much, Dr. Kinney and Dr. Park for the introduction. And it's wonderful to have whoever joined us today. And I will hope that you'll be joining us for the next, for the remaining lectures throughout the year, a presentation about this topic. We're really hoping for a very interactive type group of discussion with researchers and farmers and industry people, and it would be a very nice opportunity to discuss advances and related to cannabis. I'm going to talk to you about some of the research in my lab dealing with trying to optimization of cultivation, and specifically I'll be showing some of our recent results for effects of mineral nutrition, lighting condition, and the plant structure. First of all, we know that after a long period of not being able to conduct any research, research changes in regulation finally and thankfully allowing us to start to conduct research. In Israel, we were the first country that actually allowed legal research since cannabis was declared in 2016 as a agricultural crop, which is a legal definition that also allowed us funding. And at that time, when I was starting, and I was the first scientist to be licensed to work with whole plant cannabis in Israel, and we went into the greenhouses, and there was a lot of problems. On one hand, there's a lot of traditional growers that has a lot of experience and supposedly knew how to grow it, together with 6 companies in Israel that were growing, 6 growing companies that were growing. But we were faced with enormous questions that there was no science-based information at all to be found about. So how do we deal with this cannabis, marijuana, that was known to be, it was grown traditionally for thousands of years, and now we have to grow it for modern cultivation, either for medical or recreational use, without any information that was accumulated to other crops. In comparison, if we would compare the amount of science-based information that was available at the time or still is today for crops such as tomatoes or citrus or lettuce to cannabis at the time in 2016 or 17, when we would search, probably we would find close to 0 publication in comparison to tens of thousands of publications for every other plant. So it's a challenge. And it became immediately apparent, which still is the case today, that there is need to develop innovative agri-cultivation technologies to support this booming cannabis industry. And I will have to say, to begin with, that the challenges that we're dealing with cannabis are really unheard of for the agronomic, for agronomists, researchers, or cultivators, because we are growing, we are growing a plant that eventually goes into the pharma industry, and the level of standardization, both in terms of chemical, in terms of safety and contamination, has to be incredibly high, which is something that modern agriculture have never faced with before with any other crops. So what are some of the challenges that we're still facing? Some breakthroughs were made and are increasingly being made today. And the 2 main things, are to optimize for yield quantity, and of course, chemical quality, which is even more important at this stage than yield quantity. And today in the talk, I'll show you some of results from my lab for using mineral nutrition, fertilization, to address those issues, light quality and light intensity, and plant structure, pruning, or plant architecture. All these are geared, of course, for chemical standardization and specificity in chemical composition. And we're also conducting in my lab a lot of work about prevention of contamination, either microbial or heavy metal or pesticides or others. Post-harvest practices are unknown. There's very little information about post-harvest practices on a science level basis. And basically any other area of plant science and cultivation needs to be addressed. So it really is a wonderful, challenging task for plant science researchers, because whatever question that we ask, basically we don't have an answer to. So there's an incredible amount of information to be learned, and it's a really a challenging task to do. So if we look at cannabis, and we know that the unique thing about cannabis is that there are more than 1,000 molecules that have a potential biological activity. And these are basically mainly divided into 3 groups: cannabinoids, terpenes, and flavonoids. And the interaction between them and their composition is what affects medical properties or recreational properties. And the thing is that we know from many plants that 2 things affect very much the secondary metabolism, the chemical composition of the plant. On one hand, it's genetic. Genetic is the potential for production. Certain varieties will have a certain potential to produce, let's say, certain concentration of cannabinoids. On the other hand, there's cultivation condition, this is what will actually determine whether the potential for production will be satisfied. So the actual production of cannabinoids or terpenes or whatever will be depending on cultivation conditions. And therefore, those two aspects has to be considered when we're talking about cannabis. And today we'll be talking about cultivation practices, but I will demonstrate that many of them for a variety of cultivars. One thing that is I realized very early in the stage, we also had very detailed record for this in 2016, is that when we look at the plant and the cannabis plant, it doesn't matter whether, which variety it is, whether we grow it as a short plant or a tall plant. There are gradients in chemical profile along the plants. So in fluorescence or so-called flowers, at the top of the plant or center of the plant or bottom of the plant, they will not have uniform chemical composition. And this is we've shown this in a number of places, and other people have shown this as well. You can, for example, look at one of our research publication, which is all of this information that I'll be presenting today is available online in scientific publications. And for example, when we check concentration in the top of the flowers and of the plant, center or bottom of the plant, we can see that there are very large gradients in some cases. So for example, for THC, the major cannabinoids, we can see that in fluorescence in the bottom and center of the plant will have much lower concentration than in fluorescence in the top of the plant. And the same thing occurs for CBD, and the same thing occurs for CBN, for CBG, and CBN, which is a breakdown of THC, actually shows a different effect. Now, this is highly unacceptable if we're dealing with something that needs to be have a relatively consistent medical compounds or compounds that have a psychotropic effect. If the patient is taking 1 gram and he doesn't know what the concentration is here and here, or if it's taking it out of the bed, the concentration will vary. This is severe. So standardization is a major issue, and also standardizing and optimizing for specific compounds are an issue. And this is really the first experiment I think that I know of that was conducted. We published it only in 2019, but we've been showing those results from 2015 or 16 already. And the first question that we asked, can we affect the concentration at all? So what we've done in this initial experiment is we've fertilized the plant. We did the fertilization. We said, okay, let's try nutrition. Nutrition is a major factor that affects secondary metabolism in plant and plant function. So we fertilized the plant by the commercial fertilization that was used at the time in the cultivation farm. We added 20% phosphorus or NPK or humic acid. Why humic acid? Because at the time, many people were saying, and until today also, that humic acid helps to increase cannabinoid production. So it was very tempting. We said, where do we start with? Well, we start with whatever seems to be very important. At least many people were saying that it's very important. What we found out that it's, yes, mineral nutrition can affect and does affect production of secondary metabolites, not always in the direction that we would want to. For example, THC, this is a total THC and THCA concentration, was actually reduced by humic acid. So did CBN, and so did CBG. So that means, but if we look at overall supplementation of higher concentration of NP and K, in this case, we also received a reduction in CBN, so breakdown was reduced, and we did receive an increase in some of the cannabinoids, for example, in CBG and in THC. So nutrition can affect cannabinoid production, and humic acid not necessarily give us the same effect that we were hoping to receive or expecting to receive based on what traditional growers were saying. But it's more complex because, for example, now if we look at the same experiment, but instead of looking only at the inflorescence of the top of the plant, the cola, so-called, the cola, we were actually looking at the inflorescence again from 3 locations along the plant, and then on the same type of treatments. And what we see here that yes, humic acid reduced the concentration, but the reduction was mainly in the top, in the cola, in the top flowers. And by doing this, the concentration was lower, but actually the standardization of the plant increased. And this again tells us that it's not only effect on specific cannabinoids that we can achieve by nutritional effect, we can also have some kind of effect on standardization in the plant. And that really told me that this is the way to go. We have to at least dive in for a while into nutrition to see what we can do in order to help quality and quantity of the plant. So the main thing is, again, was how can we affect plant performance? And I will show today the results for our mineral nutrition studies, and some for our light intensity and some for plant agricultural studies. And these are all based on the fact, as I said before, that we know that either environmental conditions or cultivation conditions can have a direct effect on secondary metabolism and therefore on production of cannabinoid terpenes and flavonoids. And on the other hand, there can be an indirect effect, but they can affect physiology, plant function, or morphology on yield. And this will have an indirect effect as well on secondary metabolism. And because, of course, cannabis is a short day plant, so there is a under long photo period, it will go through vegetative growth. So this will be during the veg stage. And under short photo period, it will go if flowering will be induced. So we get generative or reproductive growth. So our experiments were divided for the vegetative growth separately from the reproductive growth, and later on also for different stages of the flowering. And I want to start by showing you some of our results for nitrogen. Again, all this is published and you can find it in the reference that I always put at the bottom of the screen. And this was done by Avia Salona, a Ph.D. student in the lab. And we did it for several cultivars. We're showing here just for one cultivar at that time. And we started by applying very relatively large range of nitrogen concentration from 30 ppm all the way up to 320 ppm, milligram per liter. And what we can see immediately, just before looking even at the graphs, that we can see that under 30 ppm, the plants are very small and undeveloped. And 80 ppm is kind of borderline, and 160 looks great. We can also see this from the way that the leaves look. And if we look at the function of the plant or biomass, for example, if we look at leaf biomass, so it will increase all the way up to 160, and so does other stem and roots as well. So for this and many other physiological parameters that we measured, and mineral accumulation for whoever's interested in exactly which concentration are in the roots and the leaves and in the flowers, and so no flowers, even the stem, you can go into the publication to take a look at it. So what we recommend, at least from those experiments, that what we came out is for the vegetative stage, 160 ppm seems to be the optimal range. What happened though for the cultivation, for the flowering stage? For the flowering stage, again, what we did was used a wide range of concentration, the same concentration from 30 to 320 ppm. And what we can see is if we look under 30 ppm, the plants were reduced in weight. There was chlorosis, which demonstrates nitrogen deficiency. At about 160, the plant looks great. And above this, we're not sure that there was any a positive effect in terms of physiological function, even negative effects. So in terms of when you look at biomass development, growth, and yield quantity, the way to go is 160. However, you know what? When we actually checked secondary metabolite production, the plants that gave us the highest concentration of most cannabinoids and most terpenes were those suffering, hungry, nitrogen-hungry plants that received 30 ppm nitrogen. And this is kind of confusing, but we only many figures, but we only want to look at the trend. So this is some of the cannabinoids that we measured. We can see a lot more in the manuscript, and we can see there the concentration of nitrogen, which increases in this direction. And let's for example at THC. THC reduces with increase of nitrogen concentration. CBD also reduces. CBC also reduces, and CBG remains stable until about 160 and reduces above it. And for many of the terpenes, we have a definition in a paper between monoterpenes and sesquiterpenes and others. For many of them, we again see the higher concentration at the lower concentration of nitrogen. Sometimes all the way up to 160, we receive higher concentration of terpenes. So definitely under stress and the nitrogen deficiency stress, we get higher production of cannabinoids on a tissue basis. Still, when we talk about the whole plant basis, and it's again depending whether someone is looking at the quality of the bud itself, quality of the inflorescence itself, or for extraction purposes, and then obviously we have to look at production per plant or per meter square, or per meter square. Okay, I want to show some results for potassium. Potassium was also done by Avias Saloner, a PhD student in the lab. And again, we checked 5 different treatments of potassium. Here I want to show result for 2 varieties. So this would be result for 1 variety and for a second variety. And we checked from 15 ppm all the way up to 240 ppm. And it's again apparent, this is a vegetative growth set during veg, that 15 is on the low side, 60 is kind of borderline, and from 100 and 175 gave us the best biomass production and physiological function. 240 was a little bit above, and this also can be seen based on the leaf. Based on the leaf, and a lot of the physiological function you can turn to the manuscript to see function under veg. What happened under the reproductive stage during flowering? Again, at the same rate, 15 to 240, and we can see that the leaf are chlorotic, the plant are chlorotic under 15 and 60, and again, somewhere around the range of 100 will probably be the best concentration, and no need to go above it. We even get some negative symptoms when we go above, as you can see here from the leaves, that sometimes they will actually get negative effect under higher concentration. But what happened again to cannabinoids, similar to what happened in nitrogen, that under the deficiency condition of 15 or 15 ppm or even 60 ppm, where the plants were smaller and suffering from deficiency, we actually received the highest concentration of cannabinoids. So, for example, if we look at this, THCA was reduced greatly. It was much higher under deficiency condition than under higher concentration, and CBDA did the same thing. And THCVA, that this variety has some THCVA as well, was again higher under deficiency conditions, and same thing occurred for CBCA and CBGA. So again, under potassium condition as well, going too high, meaning compromising concentration of cannabinoids. The same thing occurred for terpenes. Again, here for the terpenes, some of them were not affected, some of them were affected mildly, and some of them were affected only after a certain concentration. So the thing is, it probably has to do with the profile of the terpenes that the specific variety that we are growing has, and also the interest of whether to increase or stabilize for terpenes concentration as well, that we have to be careful about pushing too much potassium, too much nitrogen, and hitting the inhibition of reducing some of the production of the secondary metabolites. Just to show you very quickly about the biomass for this potassium experiment. So if we look at the yield, this is the flower yield, the inflorescence yield. Again, we see that of course lower production under the lower deficiency, but from 100 ppm and above, there was no, in the 2 varieties, this is one variety and this is another variety, there were no statistically significant differences. So again, from our experiment with the varieties that we work with, there's no reason to go above 100 ppm, especially that we are risking reducing cannabinoids and terpene production. Some words about phosphorus. This is Sivan Shiponi. Sivan is a master student in the lab, and we've done again, we checked 5 concentration of phosphorus. Now for phosphorus, we know most everyone is saying cannabis is hungry for phosphorus, especially during the flowering stage, during the reproductive stage. So this is just to show you two varieties of medical cannabis. This is during the vegetative phase. We checked a range between 5 ppm to 90 ppm. And here I want to say that normally when I started a couple of years ago, and everyone was saying cannabis is hungry for phosphorus, and some of the concentration that at least the blogs were saying you have to go several hundred ppms. This was like shocking, because normally in agriculture, in soilless cultivation in pots, we will probably go a maximum of 30 ppm, often 10 ppm or 15 ppm. So even daring to do an experiment that will go up to 90 ppm is amazing. And in this case, cannabis is indeed unique in a way. When we look at the range from 5 to 90 ppm phosphorus, we again see that 5 was a little bit on the low side. 15 was kind of borderline, and we really saw a very wide optimum range for potassium nutrition in cannabis. We definitely did not see any toxicity, but also we did not see any benefit to going above 30 ppm. I want to remind you that this is giving constant concentration throughout the flowering period, other than the flushing, other than irrigation without the nutrients at the end. And for I will not have time to show today, but we've done a lot of experiments, a lot of studies checking how much phosphorus and how much nitrogen, all the micro and micronutrients are being taken up throughout the cultivation period. And this manuscript will be out soon. And I think that some of the results will be surprising, because we're all expecting cannabis to devour a lot of phosphorus toward the end of the cultivation period. And the question is this something that's required for optimal growth and for optimal production? So these are the results that we did for the flowering period. This is for two varieties of cannabis, medical cannabis. And again, we've used from 5 to 90 ppm phosphorus. And again, what we've seen is very similar to what we've seen for nitrogen and potassium. The actually highest concentration of secondary metabolite was on these plants that suffered from phosphorus deficiency. And when we look at the yield, so so we basically see this is like the total biomass of the plant. This is yield, dry weight of flowers, of inflorescence in the two varieties, and we can see that above 30 ppm there is no increase in biomass, in yield. Same thing here, and above 30 ppm we're not statistically increasing in yield. So yield, the weight, the biomass of the flowers were not affected in concentration above 30 ppm, or maybe a little bit more, maybe 60 if we are kind of borderline on the statistical significance here. But what happened to the cannabinoids? Look at the dramatic reduction in cannabinoid concentration. So if we look at CBDA, this is in the hungry plants. In all those curves, again, the concentration of phosphorus will be in the bottom. So 5 will be deficient concentration, and all the way up to 90. And this is for the different cannabinoids. So for the two varieties, so this is one variety and this is another variety. And for example, when we look at CBDA, it's CBDA production, we see a large reduction when between the deficiency concentration and the highest concentration. So if we go from 5 to 90, we can see considerable reduction in cannabinoid concentration. And this is the same for THC concentration, and this is the same for CBDVA concentration, and much less of an effect for CBC. So again, which concentration do we choose? It really depends on the growers or the grower's preference. If we're going for larger biomass, then probably we're going for a middle range kind of concentration, 30 ppm or maybe slightly above it, up to 60, I would say, no longer. But by doing this, we're getting higher biomass on account of lower concentration of cannabinoids. So this needs to be taken into consideration. And of course, we have no intention of growing plants that suffer from deficiencies, but we have to learn that this is what we are developing now. We are developing breakthrough technologies in order to fool the plants to think that they are under deficiency or under some kind of deficiency of nutrient stress condition without actually compromising plant development. I think I'll just show this very briefly. That this is when we look at parameters of plant function, for example, photosynthesis, which would be lower under the deficiency condition, but all the range of 30 to 90, the same function, the same rate of photosynthesis, the same rate of transpiration, the same rate of stomatal conductance. That means that in terms of plant function, there's no reason to go above those concentrations. I want to show some of our results for light concentration, and then I will finish with talking about plant structure. So what about light? I mean, when we talk about light spectrum, it's a big deal in the cannabis industry those days. The type of lens that we use. Obviously, if traditionally high pressure sodium HPS lamps were used, but there's a large shift those days into using LED lighting, mainly because they require less energy, so it's cheaper to do. But there's some thoughts also about the fact that LED lighting might be actually better for plant cultivation. And I want to show you some of our results, in again a paper that we published last year. So we checked actually four different treatments. We checked the HPS. We checked an HPS lamp versus 3 LED spectrums. One of them would be LED that was rich in white, so it was full spectrum with roughly one to one blue and red light, and then two LED lamps that gave 1 to 1 and 1 to 4 blue and red light. And we checked 3 different cultivars. In this case, this was a high THC cultivar and a balanced THC and CBD cultivars and a high CBD cultivar. And the only thing that I can say 2 things. One of them is that obviously light quality affects the profile of secondary of cannabinoids, so it affects cannabinoid production. There's something which is less pleasant to hear is that at least in our study that we actually found differences between varieties in the response. And why is it not that great? It would have been better to say that for all cultivars of cannabis, if you want to get a higher CBG concentration, then you need to switch into lead, this is what we've seen here. If we look at the CBG for, in this case, for all 3 varieties, we're actually receiving lower CBG than for the 3 lead treatments, which had again 1 to 1 white light and 1 to 1 and 1 to 4 red blue to red ratio. However, when we look, for example, at CBD, we actually received the opposite for one variety. One variety under HPS gave us much higher concentration of CBD. In the other varieties, another variety did not have any significant effect to the light, and in one variety, actually CBD was slightly lower. So how does CBD respond? Apparently, there's differences between varieties, and we've seen this also for many of cannabinoids and terpenes. So there is variation in the effect of light spectrum. Some uniformity that appeared was in plant development, morphological development. And this is something that's quite well known for many plants. We know what effect the blue light is supposed to have, and what more enriched in red light will have. And plant morphogenesis is very well studied, and in this respect, we did not see any different things for cannabis, but the effect on the secondary metabolism is challenging because, at least from our study, the varieties respond differently. That means that every grower will really have to know the specific variety that he's working with in order to optimize for light spectrum, light quality. I want to end by showing you some of our studies into plant architecture. The variability that exists between growers in terms of cultivation techniques and plant shaping is enormous. Some growers will grow very small plants, more dense. Then some plants will tend to grow much larger plants. And also in terms of the shaping of the plants, we know a removal of secondary branches, we know leaf removal, we know pruning one topping or 2 toppings, variety of things. What we've done is we did a series of experiments. These are published in 2 papers. And what we did, we took those plants, and these are 2 varieties. For example, we've done this for much more. You can look at the papers, but this is for 2 varieties. And this is the control plants. We didn't touch them at all. In these plants, we did leaf removal. In these plants, we removed everything from the 3rd bottom of the plants and in this treatment, we removed branches from the bottom of the secondary branches from the bottom of the plant, including defoliation. Here we removed only the secondary branches, or only the secondary branches. Also, the primary branches was too drastic, and we did 1 topping and 2 toppings. So this is 1 pruning or 2 pruning. And what were we looking at? Obviously, amount of yield, biomass of the inflorescence. And the second thing, most importantly, is the standardization, because the idea here for removing leaves, for example, removing some of the branches, is to allow more light to come in, as well as to allow more air to move in to reduce humidity to help with sanitation. But what we've done in all those experiments is we actually selected 5 different locations. We removed flowers and analyzed them for chemical quality from 5 locations, from the colli, the top inflorescence from 2 different locations at the top and at the bottom, and then inner inflorescence from 2 heights of the plant. And we were looking to see whether we get any increased in standardization. This was, of course, followed by measuring light intensity, the amount of light that goes through the plant. What will happen here? Obviously, more light will go here than under more dense plants. And we checked for standardization. And this is in a nutshell, as we say, what we've seen is that first of all, most of the inflorescents that were on the top of the plant and exposed to light had similar concentration, not surprisingly. But bottom inflorescence, which are hidden, had reduced concentration of cannabinoids. And some of those treatments, the treatments that allowed more light to move in or more airy branches, usually let more light goes in and improve standardization by increasing concentration in the bottom of the plant. So that tells us that now the question is, should we go into very drastic type pruning that some growers, many growers are telling more and more to do? Shall we go into increased lighting at the bottom of the plant? We are dealing now, right now, what we are setting up to start probably within a month, and lighting in canopy lighting experiment that may solve the problem, because the issue is that all those pruning are incredibly labor consuming. If we can do this by any other simpler means and increase standardization and make those inflorescences to become larger, it obviously will be better. Just one word about this, and with this I will finish, is that we also did, obviously when we're dealing with plant pruning, the density of the plant stand is critical. How many plants per meter square we have, because the more plants we have per meter square, the more dense the plant is, and this will affect the need for plant shaping, for pruning. So we've done a lot of work on this as well. Obviously, the issue, the more denser we go with the plant, we have to be more careful with plant structure. I want to finish by saying that in terms of breakthroughs and innovations, what's needed? First of all, we need to develop, as we've said, agrotechniques for improvement of cultivations. This is a plant that has not been studied as intensively as other plants. The information that we have on it is very basic, and for some other crops, the information that we don't have now, we know already from the 60s for other plants. So this is critical. But we have to develop advanced technology for directing the chemical quality. It's not a tomato that we eat in a salad. It's something that people consume for medicine, or if not as a medical product, for life quality product. The chemistry has to be precise, and this requires advanced agronomic techniques. And of course, sometimes we need, or in the future, where more medical information will be available about what specific profiles we need for specific medical indications, we will probably need, or we already need, to control specific chemical quality. So to conclude or to summarize, I hope that I've convinced you that environmental conditions have a very large effect on the chemical profile, on secondary metabolite profile. Mineral nutrition has a very considerable impact on secondary metabolism in cannabis, and we have a power to regulate to a certain extent, or we need to understand the effects in order to make sure that we are receiving and growing what we are hoping to grow. And also, we have to remember that we've seen it for some of the nutrients, yield quantity and chemical quality is not always affected the same. Sometimes then the chemical, the secondary metabolite concentration can increase where the biomass decrease. And I have to say that although I have shown this for NP and K, for nitrogen, phosphorus, and potassium, where we've seen that for all those 3 under deficiency symptoms, we had higher concentration of cannabinoids. This is not the case, and suggest that you wait for for our next publication that will be on magnesium, and there we actually see the opposite trend, that under deficiency we see a reduction in cannabinoids. So it's not just a matter of stress response, it's something nutrient specific. And overall, there's an urgent need for science-based information in order to develop this magnificent cannabis industry. I want to thank, obviously, my group. It's very active and happy and dedicated group. It's very difficult to work on medical cannabis, having to be bound by all those regulations and continuously being careful. But it's a very happy type of work, I would say. And some of my collaborators, Dr. Molly Sachs from the Extension Service in Israel, that give us a lot of input as well on nutrition and conservation and how to build those a very complex nutrient solution for the experiment. And Dr. Jonathan Gorelik was a collaborator for our humic acid experiment. And the funding for this came from the chief scientist of the Ministry of Agriculture. Thank you.
Thank you, Dr. Bernstein, for that presentation. I appreciate the broad nature of the insights that you've provided over the course of the presentation. We do have a few questions that have been entered into the Q&A function, and attendees can continue to add questions as we enter the Q&A period. I also want to let folks know that we'll be putting a link to a very short survey just for attendees to provide feedback to us as we engage in this new webinar series. All right. So the first question that I want to raise is, how do you manage the post-harvest stage and room storage where cannabinoid ratios can easily change?
I will take it that the question is not specific for our experiment, but as a general question. So
well, that I can't specify, but perhaps you could address it how it's managed in your experimental work.
Okay, so I will just say that in our experimental work, we follow the same protocols that are being used for commercial cannabis. So that means that we do go through usually about 14 days of drying process. So it's a slow drying under relatively high humidity. We try to shoot for 19 degrees C, 19.5 degrees C to 20 degrees C, and so on. And once the humidity reaches kind of a desired stage, then we go into a curing stage, which is kind of an aging stage for whoever is maybe not know what it is, which is again going through and this depends on the variety, but usually we try not to go over two to 3 weeks before we analyze the material, in which we expose the plants to a cycle of opening and closing the cans and mixing. So in a way, letting the humidity inside of the fluorescence to equilibrate and for the chemistry to still continue. So we follow the exact process that's being used in the production industry. And I just want to say that we actually, because 2.5 years ago, there's no information about it other than many protocols that were conducted by many growers. So, I mean, I've visited many growers in the US and in Europe and in Israel. Everyone was saying something slightly different. And so we started 2.5 years ago a post-harvest experiment. And that actually today we analyzed, I think, the last batch because we ran out of the samples, maybe for another batch or so. And what we did is we analyzed storage under 5 different conditions. So after we finished the drying process for post-harvest, we stored them under 5 different temperatures, from very high to very low and everything in between. And we did a 1 week curing, 2 weeks curing, a 3-week curing, analyze it for the same thing, check different levels of vacuum and so on and so forth. And I'm really looking to see how this will change. And today there's also some information in the literature, very little, about the people looking into post-harvest deterioration, let's say, or changes in the profile.
Great, thank you. Another question is, what kind of inert material do you use for the studies to avoid the microbial compositions at the soil, which can affect the plant uptake?
Chad, I heard all the question except the one word. What kind of what? The very beginning of your question.
Certainly. What kind of inert material?
Oh, okay. Thank you, Dr. Feeney. Because this is a nutrient, I will differentiate between the nutrient studies and other studies. For the nutrient studies, and this is a great question again, and thank you for asking, is that for the nutrient studies, the goal is really to learn how the plant responds. What does the plant take up every week? So in all those studies, we cut the plants every week and analyze stems and leaves, so we know the uptake into the plant. So in order to do this, and in order to see plant response, we have to separate this from interaction with the soil. And that's why this question is very important, because if we were to do this experiment in more of an organic media that is very often used today, such as coconut fibers with different type of mixtures, a lot of our nutrients will be absorbed to the medium, and not necessarily the roots will see and take up what we are giving the plants. In order to do it, a media that's very commonly used in nutrient uptake studies is perlite. Perlite is an inorganic inert media. So basically, when we get those beds, we irrigate them, let's say, with some media for a day or two before we plant. Whatever you put in, that's what the plant will see. There's very, very little, if any, absorption into the media, or some things that will not come out of the media, so it's an inert media. So our nutrient studies are all conducted in perlite. It's in a way maybe similar to hydroponics, other than the fact that it's in a normal cycle of drying and wetting that we don't see in hydroponics. So it's sometimes it's different than hydroponics. So it's similar to soil. And in the other experiment, we use whatever similar media to what the growers are using, more organic-based media, obviously.
Great. The next question we have is, what is the DLI involved here? And I'm presuming that is daily light integral. Is there a specific daily light integral being used in the experimental work?
If you're talking about intensity, or when we're talking about light intensity, then we try to adhere, because of course in some of our experiments that we did for light intensity studies, we've actually found for some of our cultivation studies that already under 1300 microamps per meter square per second, we found some damage, tissue damage, which is, I know that in some other publications, people are showing also already positive responses, even up to close to 2000. We didn't find it in our experiment. We were using, in some of the experiments, we were using ambient CO2 concentration. So maybe this is one of the causes, because we were debating whether to enrich with CO2. Obviously, when we're using large indoor cultivation, CO2 is an advantage. So we calibrate for the light intensity based on this, and we usually run into something like 900 to maybe 1,000 microamps per millisecond per second. And I guess I'll stop with this.
Okay. Thank you. Another question that has come in is, given your work, how does Israeli cannabis production compare with the rest of the world? It's a fairly broad question, I think, open to a little bit of interpretation. But right. Are different techniques being employed?
Yeah, well, I will say that I think I'll answer the question directly, because I was debating, should I answer about how does Israeli production compare, or you said in light of my work. So I said in light of my work, from the calls that I get constantly for farmers, I know that a lot of our farmers, most of them, have shifted into at least their nutrient composition considerably based on the results, that we've been publishing and lecturing in many places in Israel as well, and our extension service. I'm also we started a group of agricultural advisors for cannabis and the extension service of the Ministry of Agriculture. I'm the advising scientist for this group. So we have a routine meetings and so the information, comes to the field all the time. I'm in the field many times in other places. And a lot of our nutrition have shifted, toward this. Many people have reduced their concentration based on this. So we are in constant, I think one of the things that's very unique to Israel agriculture is that there's a very, very strong bond between, we call this a triangle between research and advising and the advisory extension service and the farmers. So information flows back and forth, which is great because we hear all the time about problems and issues that needs to be studied. This is what we study, and information goes back. In terms of overall production, Israel, we can only grow cannabis in soilless cultivation, not in the field and not in soil. So the studies that we are doing are directly related, to what's allowed to be done in Israel. If in 2016 we have 6 cultivators, we have over 40 now. And the industry is growing amazingly, the same as it is throughout the world. We are very, very heavily into research, and we try to convince everyone that it's the best advantage of everyone to share information. So I think that we are seeing a small shift from the hush-hush mentality that every growers tend to think that, whatever he discovers is a very big secret that is to be kept. We believe that, if you keep information to yourself, that means that only the information that came up from one farm. If you share information with 20 farms, you receive back the information from 20 farms. So this is what's accepted in agriculture generally in Israel. We're still not nearly there in cannabis, in the cannabis production. Hopefully, maybe we'll get there one day.
Okay. You talked about standardization of chemical composition of cannabis as being very important, especially as it enters pharmaceutical use or medical use. Has there been any attempt to conduct standardization of materials post-harvest to homogenize it, to generate a final product that fits a standardized composition?
Thank you, Dr. Kinney. And again, this is a great question. In Israel, the range of products that we sell are defined by law, by the IMC, by the Israel Medical Cannabis Unit of the Ministry of Agriculture. So it is, if you look at and everything that we sell in pharmacies today. So these are not dispensaries. These are normal pharmacies. The pharmacies that wanted to go into selling medical product, and it's only medical in Israel so far, has actually to go into training, build safes, train the pharmacies. So we have a huge number of, we have participate in courses through the Ministry of Health. So I teach in courses for doctors, for nurses, for health practitioners, for pharmacists. It's all going now through the universities and through the universities throughout Israel. And so you have to have trained pharmacies that can sell those products and being sold through pharmacies. Why am I giving you this information? Because it's very restricted in what you can sell. So we have a table that define different ratios between THC and CBD, defined ratios that all the producers in Israel have to adhere to those ratios. So everything that will be above scale in THC will not have sufficient level in CBD, for example, will not be able to be sold, and so on. And so in terms of standardization, all the producers, there is a separation by law now in Israel between the growers and between the factories that pack it, and delivery and shipment is another issue, and then selling is through the pharmacies. So we have different branches that do it. And standardization actually occurs because everything has to come to a pharmacy as being packed and checked and has to be certified for every batch of plants to be matching one of those levels of standardization. And then every medical product eventually will be adhered to one of those allowed levels that we have the stickers that will say at least how much THC and CBD and CBN. So just like the very few of the cannabinoids. In terms of mixing, in order to mix, that means having to grind, right? So if we grind, it can also be into batches of cigarettes, for example, that are being sold, or into extractions. One of the three main projects that we are allowed to sell in Israel are either ground material in cigarettes or in bulk and capsules, or the flowers themselves, dry fluorescents, or extractions, which are mixed in oil. So there are no edibles or chocolates or gummy bears, non-existing. And with it comes the standardization into this. So if we're dealing with large batches, they will go into extraction. So again, for the extract, again, they will have to follow the same THC to CBD ratios as we find in the plant material, in the flowers that are being sold. Above this, so there's so the standardization occurs in the factory, but still, if the material that will come from the field will not follow into specific categories, this is not going to be possible. So whatever we go around it, we still have to standardize in the field in order to allow the pharmaceutical industry later on to pack it to the required ranges for conservation.
Great. We have time for one final question. And the last question is, since CBG in its acidic form, CBGA, is the primary precursor of all phytocannabinoids, wouldn't that suggest that any form of biotic or abiotic treatment or stress that impacts the production of CBG subsequently reduces all secondary metabolites?
Thank you, Dr. Kinney. This is, again, a very good question, because whenever we see in my lab, we finish analyzing a profile of secondary metabolites, and we see an increase in CBG. The first thing says, wow, increased production of secondary metabolites, right? On the other hand, we say, oh no, maybe what we did is just stop the enzymatic transformation from CBGA into CBC, CBCA, CBDA, or THCA. So when we analyze for plant material, we see a temporal one-stage view. And if we see an increase in THC, we have to try to look at what happens with other major cannabinoids. If they are greatly reduced, maybe that means that CBGA is accumulating and there's no enzymatic transformation. But if we see an increase in all, then we see, okay, then we probably increase the production. So it's all, it's difficult that we see temporal one-point thing from the analysis, and we're trying from this to project on overall production. And for this, of course, we have we analyze different location on the plant, we analyze different stages of plant development. Often in our experiment, I didn't talk about it today, we analyzed at the milky trichome stage, clear trichome stage, and amber trichome stage at different stages in order to try to get an idea of the dynamics. Thank you, that's a great question.
Good. Well, that was perfect timing. We have just landed at the end of the hour. Thank you, Dr. Bernstein, for a wonderful first presentation for this new webinar series. I hope folks will join us again next month for the second webinar series, or second webinar in this series. You can find more information about the webinar series on the ICR's website, and we will certainly be putting out information through our email distribution list and through social media. Thank you, everybody, for joining us today.